is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell nucleus. The information in DNA is stored as a code made up of 4 chemical bases: adenine, guanine, thymine, and cytosine. The order or sequence of these bases determines the information available for building and maintaining an organism. DNA bases pair up with each other, A with T and C with G, to form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar, and phosphate are called a nucleotide. Nucleotides are arranged in 2 long strands that form a spiral called a double helix. An important property of DNA is that it can replicate or make copies of itself. Each strand of DNA in the double helix can serve as a pattern for duplicating the sequence of bases. In humans and almost all other organisms, there are only 4 chemical bases. Research has been done to try and expand the genetic alphabet by creating chemical bases in laboratories, making these unnatural bases. Research into unnatural base pairs happened in an American laboratory in California in the Scripps Research Institute, and the results of their research were published in 2008. This research opened new opportunities and created innovation in the field of biochemistry. Typically, the design and development of unnatural base pairs have relied on the theories of molecular recognition. However, in this experiment, 2 screens for efficient Kf-mediated synthesis were used. One, which looks for extension, and another, which looks for synthesis and continued primary extension. This was done to identify novel candidate base pairs. The strongest candidate was found to be the pair formed between DS-ICS and DMMO2. This is an hydrophobic base pair. Further research showed that the base pair is limited by the favoring self-pairing of DS-ICS to DS-ICS, likely due to the interactions between the large aromatic rings. To solve this problem, they altered the pair using activity relationship in previous studies. This resulted in the production of base pair D5-SICS to DMMO2. This made the pair highly selective and have little to no effect on any part of the replication process. After comparison with other previous candidate base pairs, D5-SICS to DMMO2 pair has more fidelity and efficiency. Following the discovery, the next thing was understanding the origins of the remarkable properties of DSICS to DMMO2 and D5SICS to DMMO2 base pairs by analyzing the different relationships, and the group made initial progress towards this goal. The structure-activity relationship, along with the growing literature regarding hydrophobic base pairs, begins to suggest design principles which will aid in the development of future unnatural base pairs. The group intends to continue to search for modifications that optimize the D5SICS to DMMO2 pair, also with the expectation that this work will tell us the fundamental principles underlying the storage and retrieval of genetic information, as well as ultimately culminate in a fully functional unnatural base pair. Since 2008, research into the development of the uses of unnatural base pairs has continued, and research into this area has increased. Just last year, a new development was made by a Japanese team. They published a paper on generation of high-affinity DNA aptamers using unnatural base pairs. One of the researchers also said, if we can add in new bases into DNA, we can make proteins containing more than 20 amino acids. We can perhaps apply it to protein synthesis with completely new amino acids, or even creating new organisms with 5 or 6 different bases in the DNA. There are many, many possibilities.